break was a little bit shorter. I hope lunch will be more relaxed. But you know, we started late, and in every bad thing there is always some good, or I don't know even if good, but some way that we can manage. So unfortunately, Professor Sarah Harper from Oxford couldn't make it. She sent her video. So we have, I hope we'll have the time to show her video, but at least we were short in one lecture, so I hope we'll try to manage the time through lunch and finish on time with our panel. So, therefore, I'm sorry for pushing you and asking you to shorten a bit and to come back, to shorten the coffee breaks and maybe also the lunch. But it's my great privilege and our great privilege and honor to have with us Professor Alan Leshner, who was with us last time both at the Dead Sea and at the Healthy Aging this year again, and I thank him very much for that. Professor Leshner is really, I wouldn't have time to go through all the stories, I know, but it's very interesting because, as I said, as we grow, we have more time to do things, and you did a lot. So he was the Chief Executive Officer of the AAAS and the former Executive Publisher of Science, and he's still working very hard. He's just writing a report now on drug addiction, and therefore we couldn't change, and he had to be here right today. So if you're asking questions, I hope he will tell you more about his activities, but now I want to invite him to chair this session. Thank you.
I don't know if I merit applause. I'm really very happy to be here. Thank you, Mira. Thank you for organizing yet another terrific conference. So I know how dangerous it is to get between people and lunch. So I just want to warn the speakers, I'm actually going to keep you to the time. I have this electronic watch. It's an Apple iWatch, and I'll set the timer on it. And when it goes, beep, please stop. But let me say that a conference like this, that it's as diverse as it is really both attractive and hopefully a source for optimism. Those of us who are aging are ever hopeful that the rest of you will find ways to make that process easier, to make it more comfortable, to make all of us healthier as we age, and I very much like the idea of talking about healthy aging. I recently had a very frustrating experience of chairing a study for the U.S. National Academy of Sciences and the National Academy of Medicine on the prevention of dementia. The answer was, don't get your hopes up. Basically, when you apply the same standard of science to the prevention of dementia, Alzheimer's, cognitive decline, that you apply to everything else in science, that is to require randomized clinical trials in order to answer the question, basically nothing works with any surety. I can tell you the three things you can try to do that will help. The most important single thing, not surprisingly, is to control blood pressure during the middle ages, that is, while you're in middle ages. If you control your blood pressure, the probability of having stroke is significantly reduced, and therefore the probability of developing dementia is reduced. Physical activity, pretty good. Probably have to do more than any of us could tolerate. And cognitive activity works to some degree, but sadly, all of those brain games that people sell, save your money. With that optimistic note, let's hope that over the course of today we find some things that will actually help improve our aging. We're going to start this session with a discussion of probably one of the most important topics for eventually getting control over cognitive decline, dementia, Alzheimer's disease, and that is the search for biomarkers. And I can tell you that the US NIH has a large initiative around dementia, Alzheimer's biomarkers. It's been making some progress, and I'm very hopeful that Professor Ilana Gozes from here at Tel Aviv University will give us more hope. Thank you.
Thank you very much, and it's a very tough act to follow after the excellent lectures that we heard this morning. So I'll try, and also my time is limited, so I'll try to be fast. So I'm speaking about biomarkers for cognition, aging, and Alzheimer's disease. And as you know, Alzheimer's disease is the most prevalent aging disease, and the major things that disappears in Alzheimer's disease is cognition. But as a woman, I always like to speak about what I do. So like 20 years ago, we started looking for proteins that protect the brain against disease and injury. And we discovered a new protein to science here in the basement of the Sackler Faculty of Medicine, and we called it activity-dependent neuroprotective protein because it protects nerve cells against blockade of electrical activity. The brain won't act if it is not electrically active. And so in short, ADNP. And then we knocked it out in mice, and we found that it's a no-brainer. If you knock ADNP out, there is no brain. So the neural tube doesn't close. So it's very important during development. And development and aging are kind of the two sides of the coin, like with the vaccines. And you need both, actually, to be operating well. And sometimes something that goes wrong during development is the same thing that goes wrong during aging. And in both cases, you have cognitive impairments. So ADNP actually, based a little bit on our studies and also with the solving of the human genome, ADNP was found to be mutated de novo during pregnancy in children, in embryos, and then it causes autism, but not only autism, the 100% of children with a mutation in ADNP suffer from intellectual disability. So it's a gene that is important for brain formation in the mouse, and also for cognition in the mouse, and also for cognition in human beings. In children, if it's mutated, the child is intellectually disabled. And sometimes they don't even talk, the children. So this is the ADNP syndrome. So now there is an ADNP syndrome. It's a very rare syndrome, but it's one of the lead genes that cause intellectual disability in children. And we are, of course, working on that because ADNP is my story. And the prevalence, as I said, it's not very high. And the prediction is like 20,000 patients worldwide, but still it's one of the leading genes that can cause this. And when I was a child, nobody knew why children are intellectually disabled or autistic. And now we know that there are genes that are leading this situation. But if you look at the aging, and I said there is a connection, so if we measure, and this was done in collaboration with the group in Harvard, if we measure ADNP in the serum of patients, not of patients, of elderly people, what we find is a correlation between the IQ test results and the amount of ADNP. So like in the children, if there is a mutation and there is less active ADNP, and then the children are cognitively disabled. Similarly, in elderly people, they are cognitively disabled if they have less ADNP. So it's in both sides of the coin. And we also devised a very small peptide out of ADNP that can provide protection. And what we saw, if we knock ADNP in the mouse, there is no brain. And in the human, it is associated with cognitive ability. If we knock ADNP in the cell, in the nerve cell, we actually lose processes. So this is the normal cell. You see the long processes. In tissue culture, you knock out ADNP, the processes are lost. If we do like oxidative stress, which is associated with aging in nerve cells, and then we add this very small fragment from ADNP, it actually brings back the processes. And we know how it does it. So now I don't know how many of you are biochemists, but we know how it does it. It binds to the microtubules. So microtubules are like cylinders within the nerve cells that maintain the nerve cell stability. So it binds to the microtubules, to the end of the microtubules. We know exactly how. And then it carries the microtubules' dynamicity. So it's very important for the microtubules to be dynamic. So it's not like our brain is not like it's a building, but it's a moving building, and we need plasticity. So that's how it works. And by that, it actually causes a synaptic formation. So you see, this is the nerve cell without, you see the nerve cell with many processes, without the addition of the small fragment from ADNP that we call NAP, or davunetide or CP201 if I want to mix you up completely. And this is the dendritic spines or the synapses that are made as a cause of the addition of NAP. So if you have less ADNP, or if you have a mutation in ADNP, you will have less synaptic plasticity, so less cognition. So this is a clear cut. And we showed it also in animals. So we have now animals that are labeled with green fluorescent proteins. So we can see, and the nerve cells are labeled, and we can see the dendritic spines. We can see the connection. These are this membranous invagination from the process of the nerve cell, or the axon as we call it. And you see that when the animal has less ADNP, there are less processes, so there are less connections. And the animal is also cognitively impaired. When we add our small peptide back by injection, you see that we actually bring it back. And here, if we look, again, this is again in the animals, we look at the animal in terms of excitatory synapses. So these synapses are very important for cognition. And what you see here, it's night and day. This is the normal animal. This is the animal that has less ADNP. And this is the animal that was treated with our compound. And this is the quantitation. So this is the glutamatergic synapse that is going up. And this was published in 2019. So I'm telling you, hot off the oven. And so what happens with Alzheimer's disease? In Alzheimer's, so aging per se actually is associated with a little bit of death of nerve cells. So all of us sitting here, we don't have so many, I hope, hopefully we have all our brain intact, although we have a little bit neuronal cell death. In Alzheimer's disease, this process is a bit accelerated, and we lose nerve cells and we lose synapses, and that's why the cognition is lost. And what is going on together with it is these microtubules that I was talking about a minute ago. They are also deteriorating during Alzheimer's disease, and there are two pathologies that Dr. Alzheimer saw more than 100 years ago. He saw amyloid plaques and neurofibrillary tangles that are made of the tau protein, and I always like to speak, when I speak to lay audience, I say Tel Aviv University protein, but it's actually, the tau protein is actually associates with the microtubules and stabilize them and also increases their dynamicity. So what about tau? So this is a work that we published 2 years ago. What we showed is that ADNP is not only associated with the dynamicity of the microtubules, but also the small fragment from ADNP that increases ADNP activity, which we call NAP, actually enhances by interacting with the same protein that is at the end of the microtubule. It enhances by 20-fold tau binding to the microtubule shaft. So then it won't fall apart and actually cause these neurofibrillary tangles. So this is the connection, and you see here the increase. So the increase is measurable. And a paper that came out yesterday, and I couldn't have talked to you about it if it didn't come out yesterday, just published. Actually, there are two, globally speaking, there are two types of tau. So it's one gene, but it is alternatively spliced, and then you have 3-repeat tau, so 3-repeat binding to the microtubule or to tubulin, which is a subunit of the microtubule, or 4-repeat. The 3-repeat is more associated with development, and it is associated with dynamic microtubules. The 4-repeat tau is associated more with aging. In the animals, it's black and white. The young animals have 3-repeat, the old animal have 4-repeat. In human beings, it's not exactly black and white. The 3-repeat is associated with brain development, but there are 4-repeat dementias or 4-repeat tauopathies. And then all the tangles are 4-repeat tau in Alzheimer's disease, it's both the 3-repeat and the 4-repeat, meaning that if you are designing some drugs to interact with these neurofibrillary tangles, you have to be specific because they are not the same. And what we showed is that, and I'm cutting a long story short, what we showed is that the tau 4-repeat and the 3-repeat are differentially phosphorylated, so they undergo a change that will make the association with the microtubule different. And our small compound actually enhances more the activity of the 3-repeat, which means that it won't be good for 4-repeat tauopathies, but it will be very good for development, and it is associated with brain formation, and also for maybe Alzheimer's disease. And so the small peptide that we derive from ADNP has been in clinical development, and we call it also CP201 or davunetide. It shows safety. And here I show you just a small clinical trial where it actually enhanced learning, enhanced short-term memory, meaning remembering a picture that was shown to you after 12 seconds again. So this is a short clinical trial, and we are thinking about further developments. So now more about the markers. So we are doing biomarkers. ADNP is not the only protein. As you saw, there are 23,000 genes, and many of them are expressed in the brain. So one way we are looking at biomarkers is, we can't take samples from the human brain, so we take it from the blood. So we are looking at peripheral blood and at lymphoblastoid cells. So it's lymphocytes that had been immortalized. And we did a complete Affymetrix array. So we looked at all the genes that are expressed. And then we discovered a gene that is called RGS2 that is, so what we did is actually we looked, and when I say we, it's together with Edva Hadar and David Gurevich, Professor David Gurevich in our department, and this is a PhD project. So we looked to see what is changing in these lymphocytes as a consequence of sensitivity to amyloid beta, which is in the plaque. And we saw this specific gene that is changing both in this is the low sensitivity to the amyloid beta, is changing both in the lymphoblastoid cells and also in patients' blood. And so this paper was published, and this is actually a predictor of the sensitivity of the person to Alzheimer's disease. So this is one biomarker, and if we know more of these biomarkers, we actually can screen the population, and then once we have a drug for treatment, like my fantasy over there, and others, then we can continue. So this is like one biomarker. The other thing, that the other side of the coin is, there are people, I'm sure everybody in this room, that reach the age of 100 and they are cognizant, they are fantastic. I always gave this, like President Perez, I always gave him as an example because he was giving talks and everything, and as if age went to somebody else. So the question is, what is the difference between these centenarians and the rest of the people, because that also would help us both in drug design and also in like screening the population. So we looked at that as well. And there is a protein that is known to be associated with aging and healthy aging in worms. So it's called SIRT1. And what we found here is that SIRT1 is much more expressed in the women age over 100 as compared with Alzheimer's patients. And also we looked, so this is also something that is now very modern, because our genome expresses mRNAs, messenger RNA that makes protein, but also there are many other, most of the DNA, we don't know exactly what it does. That's the truth. So we are looking at a very small fraction of the DNA. So now there are also regulatory elements within the DNA. Some of them are called microRNA. So we know microRNA that regulate SIRT1 and they negatively regulate SIRT1 sometimes. So you see there is both the SIRT1 and the microRNA are regulated with the centenarians. And they are also correlated with cognition. So the less SIRT1 the person has, the less cognizant the person is. And this was published in scientific reports, recently, and it was the editor choice, and also my student yesterday got a prize for this work, the best prize in biological psychiatry in Israel. So I'm actually much faster than I thought. So I got a prize for the discovery of ADNP by the mothers of the ADNP children and global genes in the United States. This is my laboratory, and we are the ADNP lab. And Coronis Neurosciences is developing this small fragment from ADNP that we call CP201, originally for the ADNP syndrome. So we've been to the FDA, and this is my lab. And thank you very much, and you don't need to get me off the stage.
Wow. Okay, that was great. So, wait, these are the people always ask. Anybody else first? There's a phenomenon, it's called must ask. So, there's one.
Yeah, I
Good.
got one.
Okay.
Okay. Loud.
Let me kind of phrase this.
Short question.
So because of it is a peptide, so it has a short half-life. So not as short as many other peptides, but still short. It is like half an hour to like 2 hours, depending. But so that's not so short, but it's still short. So I would recommend maybe take it daily or something like that. However, we do have animal studies. So like during development, some of the changes are going to be permanent. So we do have animal studies showing that one injection would last for a long time. Yeah.
Okay, go ahead. I know, you have to. Go ahead. Okay.
So we have a publication on head injury for several publications on head injuries several years back. Yeah, it does. So that's a case where you need to give it only once because it will protect against the immediate injury. Yeah, it does. Yeah. Yeah.
Go ahead.
Yeah.
I wanted to ask you about the role of
So that's actually a very, very interesting question. So like in Alzheimer's disease, it has been shown that too little sleep actually is associated with accumulation of amyloid beta. So whenever I get up in the middle of the night and I start working, I think about it, but it doesn't make me stop working. So that's one thing. So sleep is associated also with the ADNP deficient, the ADNP mutated children, so the ADNP syndrome children, they also have some problems with sleep. So sleep is associated with disturbed development. So sleep is very, very important. It kind of builds our brain. And I'm not a sleep researcher, but there is a lot of sleep research going on, and it's very, very important. Also, there is a connection with the immune system. So it's really important.
Okay.
Okay. Yes, so this is something that we are doing currently. So this is not here, but yes, we are looking. We are doing like complete RNA-seq, and we are looking at everything. So that I will have something to say also next year.
I have to say something optimistic. So I was waiting to ask the question about the effect on cognitive function. Mm-hmm. And in my experience, way too few researchers who find the biomarkers of the disease then go and correlate it with cognitive function as opposed to correlating. Thank you for that.
Thank you.
You give me optimism.
Thank you.
Okay. All right. Thank you very much. That was great.